We are Group 9 will explain about FTIR. Presentation for FTIR starts now. It's my pleasure to introduce principle of FTIR. FTIR stands for Fourier transform infrared spectroscopy, the preferred method of infrared spectroscopy. First of all, infrared radiation is the energy source in infrared spectroscopy. These are longer wavelengths than visual light, so they are lower in frequency and lower in energy than visual light. This infrared radiation is used to check compound. IR radiation is passed through a sample. Some of the infrared radiation is observed by the sample, and some of it is passed through. IR spectroscopy detects light absorption in the IR region of electromagnetic spectrum, which gives information of compound functional groups. Then, why compound observe infrared light? Absorption of IR light causes changes in the vibrational motions of molecules. Covalent bonds are not static. They are more like springs with wedge on each end. When two atoms are bonded to each other, the bond stretches back and forth. When three or more atoms are joined together, bonds can also bend. These bond stretching and bending vibrations represent the different vibrational modes available to a molecule. These vibrations are quantized, so they occur only at specific frequencies, which correspond to the frequency of IR light. When the frequency of IR light matches the frequency of a particular vibration mode, the IR light is observed, causing the amplitude of the particular bond stretching or bond band to increase. So, different kind of bonds vibrate at different frequencies, so they observe different frequencies of IR light. It means that IR spectroscopy can distinguish between the different kind of bond in a molecule. So, it is possible to determine the functional groups present. And then, why FTIR is good among the lot of IR spectroscopy method? Because FTIR can measure dot IR absorption of compound with fast scanning speed. It can measure all of the infrared frequencies simultaneously, unlike dispersive IR spectroscopy, which can scan individually. This difference is made from simple optical device called an interferometer. The interferometer produces interferogram which has all the infrared frequencies encoded into it. The signal can be measured very quickly, usually on the order of 1 second. Then interferogram is decoded to frequency spectrum by mathematical technique called Fourier transformation.
Hello, my name is Dane Che, and I'm going to introduce about structure of FTIR. This is a diagram of a basic FTIR spectrometer. Radiation of all frequencies from the IR source is reflected into the interferometer, where it is modulated by the moving mirror on the left. The modulated radiation is then reflected from the two mirrors on the right through the sample in the compartment at the bottom. After passing through a sample, the radiation falls on the transducer. A data acquisition system attached to the transducer records the signal and stores it in the memory of a computer with an interferogram. The majority of commercially available FTIR instruments are based on the Michelson interferometer. The Michelson interferometer is a common configuration for optical interferometry and was invented by Albert Abraham Michelson. Using a beam splitter, a light source is split into two arms. Each of those light beams is reflected back toward the beam splitter, which can combine their amplitudes using the superposition principle. The resulting interference pattern that is not directed back toward the source is typically directed to some type of photoelectric detector or camera. For different applications of the interferometer, the two light passes can be with different lengths or incorporate optical elements or even materials under the test. FTIR spectrometers can be single beam or double beam instruments. The upper diagram shows the optics of a basic single beam spectrometer. A typical procedure for determining transmittance or absorbance with this type of instrument is to first obtain a reference interferogram by scanning a reference 20 or 30 times, co-adding the data and storing the results in the memory of the instrument computer. A sample is then inserted in the radiation path and the process repeated. The ratio of sample and reference spectral data is then computed to give the transmittance at various frequencies. From this ratio, the absorbance is calculated as a function of wave number. Ordinarily, modern IR sources and detectors are sufficiently stable so that reference spectra need to be obtained only occasionally. A double beam spectrometer is illustrated in lower diagram. The mirrors directing the interferometer beam through the sample and reference cells are oscillated rapidly compared to the movement of the interferometer mirror so that the sample and reference information can be obtained at each mirror position. The double beam design compensates for source and detector drift. IR sources consist of an inert solid that is heated electrically to a temperature between 1500 and 2200 Kelvin. These sources produce continuum radiation approximating that of a black body. The maximum radiant intensity at these temperatures occurs between 5000 and 5900 1/centimeter. At longer wavelengths, the intensity falls off smoothly until it is about 1% of the maximum at 670 1/centimeter. On the short wavelength side, the decrease is much more rapid, and a similar reduction in intensity is observed at about 1000 1/centimeter. There are four types of IR sources: Nernst Glover, tungsten filament lamp, and mercury arc lamp. IR transducers are of three general types: pyroelectric transducers, photoconducting transducers, and thermal transducers. The first is found in photometers, many FTIR spectrometers, and these first type spectrometers, photoconducting transducers, are also found in FTIR instruments. Thermal detectors are found in older dispersive instruments but are too slow to be used in FTIR.
Now let's talk about data interpretation and examples applied to actual research. In organic materials, not all covalent bonds display bands in the IR spectrum. Only polar bonds do so. These are referred to as IR active. The intensity of the bands depends on the magnitude of the dipole moment associated with the bonds as below. First, strongly polar bonds produce strong bands like this carbonyl group. Medium polarity bonds and asymmetry bond produces medium bands, and weakly polar bond and symmetry bonds produce weak or non-observable bands. The typical IR absorption range for covalent bonds is 4000 to 600 per centimeter. The graph shows the regions of the spectrum where the following types of the bonds normally absorb. For example, a sharp band around 2400 to 2200 per centimeter would indicate the possible presence of a CN or CC triple bond. The functional group region number 1, 2, 3 contains relatively few peaks. These are typically associated with the stretchy vibration of functional groups within a narrow range, so we can see the existence of a specific functional group. For 1400 to 600 range is called the fingerprint region. This is normally a complex area showing many bands, frequently overlapping each other, and it is much more difficult to pick out individual bonds in this region. However, this complexity can specify substance. So once you want to identify an unknown compound, you can compare its IR spectrum with that of a known sample of the compound. If all peaks in the fingerprint region match, you have identified your compound. FTIR data has two types, transmittance spectra and absorbance spectra. Actually, with FTIR, we only obtain transmittance of the sample with changing lab number. And absorbance spectrum is calculated with above relation or log scale of transmittance. With glance, you cannot notice the difference between two data without upside down. But important point is that observance is in algorithmic scale, and this means that it is worth that comparing the concentration of the specific chemical with the intensity of its peak in different samples. Thus, transmittance data is usually used for qualitative analysis to identify functional group in unknown samples, and absorbance data is used for quantitative analysis by comparing intensity of the peak. But it's clear that only comparison is allowed with difference absorbance, and absolute amount cannot be obtained with FTIR absorbance data. Let's see some examples. There are carbonyl compound contain the CO double bond functional group. Aldehydes and ketones are isomer. In aldehydes, this group is at the end of a carbon chain, whereas in ketones, it's in the middle of the chain. As a result, the carbon in the CO bond of aldehydes is also bonded to another carbon and hydrogen, whereas the same carbon in the ketones is bonded to two other carbons. Aldehydes and ketones show a strong prominent band around 1720 to 1710 per centimeter. This band is due to the highly polar CO double bond. Because of its position, shape, and size, it is hard to miss. Because aldehydes also contain a CO bond to the sp2 carbon of the CO bond. They also show a pair of medium strength bands positioned about 2700 and 28 per centimeter. These bands are missing in the spectrum of a ketone because the sp2 carbon of the ketone lacks the CH bond. So we can distinguish between the two. Let's see the spectra of a propanol and isopropanol having the same functional group. Both spectra are quite similar in the functional group region. We can see the broad OH band and sharp CH peak, but their absorption patterns in the fingerprint region are completely different. Thus, you can know the two molecules are different, and molecules can be characterized by comparing the reference spectral data.
Here is another example that utilizes FTIR absorbance data for some quantitative purpose. In this paper, they synthesized the PT nanoparticle with cationic surfactants CTAP, which is shown in here, consists with cationic head and hydrophobic alkyl tail, and then revealed the stability of surfactants which are surrounding PT nanoparticles with changing temperature condition. They revealed two main facts by FTIR. First one is changing of CTAP IR spectra, which is bound to PT nanoparticle. Before research, they already knew that peaks at this 3 region means CH to stretch from alkyl tail of CTAP, and 960 and 760 peak means nitrogen carbon cation cat. But when analyzing absorbance spectra of CTAP bound to PT, FWHM up to peaks are at these 2 regions. greatly increased. It is because electrons at PT metals are correlated with electrons at alkyl tail. And secondly, they measured FTIR absorption spectra at various temperature conditions. From the under figure, they found that these three regions, narrow peaks are decreased. With the above results, they could infer that increase of temperature makes both free CTAP unbound to PT and alkyl tail of CTAP bound to PT are disturbed, and only cationic heads are bound and then they surround PT nanoparticles. So in summary, FTIR can answer these following questions. We can know how atoms are connected together in the unknown composites, especially their bond types and single, double, or triple, and their functional groups. Also, we can distinguish specific stereoisomers by fingerprint region. FTIR is most useful in providing information about presence of or absence of specific functional groups, and it can provide a molecular fingerprint that can be used when comparing samples. Also, it has some advantage, such as high sensitivity or quick and easy measurements, and the measurement is non-destructive. But it also has some limitations. FTIR does not provide detailed information and proof of molecular formula or structure. Thus, it is very limited in scope and must be used in conjugation with other techniques such as MS or NMR to provide a complete picture of the molecular structure. So this is all information about FTIR. Thank you for watching our video.